On August 7, 1996, reporters, photographers, and television camera operators surged into NASA headquarters in Washington, D.C. The crowd focused not on the row of seated scientists in NASA's auditorium, but on a small, clear plastic box on the table in front of them. Inside the box was a velvet pillow, and nestled on it like a crown jewel was a rock from Mars. The scientists announced that they'd found signs of life inside the meteorite. NASA Administrator Daniel Golden gleefully said it was an unbelievable day. He was more accurate than he knew. The rock, the researchers explained, had formed 4.5 billion years ago on Mars, where it remained until 16 million years ago when it was launched into space, probably by the impact of an asteroid. The rock wandered the inner solar system until 13,000 years ago, when it fell to Antarctica. It sat on the ice near Allen Hills until 1984, when snowmobiling geologists scooped it up. Scientists headed by David McKay of the Johnson Space Center in Houston found that the rock, called Alh84001, had a peculiar chemical makeup. It contained a combination of minerals and carbon compounds that on Earth are created by microbes. It also had crystals of magnetic iron oxide called magnetite, which some bacteria produce. Moreover, McKay presented to the crowd an electron microscope view of the rock showing chains of globules that bore a striking resemblance to chains that some bacteria form on Earth. "We believe that these are indeed microfossils from Mars," McKay said, adding that the evidence wasn't absolute proof of past Martian life, but rather pointers in that direction. Among the last to speak that day was J. William Schopf, a University of California at Los Angeles paleobiologist who specializes in early Earth fossils. "I'll show you the oldest evidence of life on this planet," Schopf said to the audience, and displayed a slide of a 3.465 billion-year-old fossilized chain of microscopic globules that he had found in Australia. "These are demonstrably fossils," Schopf said, implying that NASA's Martian pictures were not. He closed by quoting the astronomer Carl Sagan, "Extraordinary claims require extraordinary evidence." Despite Schopf's note of skepticism, the NASA announcement was trumpeted worldwide. "Mars lived. Rock shows meteorite holds evidence of life on another world," said the New York Times. "Fossil from the red planet may prove that we are not alone," declared the Independent of London. Over the past 9 years, scientists have taken Sagan's words very much to heart. They've scrutinized the Martian meteorite, which is now on view at the Smithsonian's National Museum of Natural History, and today few believe that it harbored Martian microbes. The controversy has prompted scientists to ask how they can know whether some blob, crystal, or chemical oddity is a sign of life, even on Earth. A debate has flared up over some of the oldest evidence for life on Earth, including the fossils that Schopf proudly displayed in 1996. Major questions are at stake in this debate, including how life first evolved on Earth. Some scientists propose that for the first few hundred million years that life existed, it bore little resemblance to life as we know it today. NASA researchers are taking lessons from the debate about life on Earth to Mars. If all goes as planned, a new generation of rovers will arrive on Mars within the next decade. These missions will incorporate cutting-edge biotechnology designed to detect individual molecules made by Martian organisms, either living or long dead. The search for life on Mars has become more urgent thanks in part to probes by the 2 rovers now roaming Mars's surface and another spaceship that is orbiting the planet. In recent months, they've made a series of astonishing discoveries that once again tempt scientists to believe that Mars harbors life, or did so in the past. At a February conference in the Netherlands, an audience of Mars experts was surveyed about Martian life. Some 75% of the scientists said they thought life once existed there, and of them, 25% think that Mars harbors life today. The search for the fossil remains of primitive single-celled organisms like bacteria took off in 1953 when Stanley Tyler, an economic geologist at the University of Wisconsin, puzzled over some 2.1 billion-year-old rocks he'd gathered in Ontario, Canada. His glassy black rocks, known as cherts, were loaded with strange microscopic filaments and hollow balls. Working with Harvard paleobotanist Elso Barghorn, Tyler proposed that the shapes were actually fossils left behind by ancient life forms such as algae. Before Tyler and Barghorn's work, few fossils had been found that predated the Cambrian period, which began about 540 million years ago. Now the two scientists were positing that life was present much earlier in the 4.55 billion year history of our planet. How much further back it went remained for later scientists to discover. In the next decades, paleontologists in Africa found 3 billion-year-old fossil traces of microscopic bacteria that had lived in massive marine reefs. Bacteria can also form what are called biofilms, colonies that grow in thin layers over surfaces such as rocks and the ocean floor, and scientists have found solid evidence for biofilms dating back 3.2 billion years. But at the time of the NASA press conference, the oldest fossil claim belonged to UCLA's William Schopf, the man who spoke skeptically about NASA's finds at the same conference. During the 1960s, 70s, and 80s, Schopf had become a leading expert on early life forms, discovering fossils around the world, including 3 billion-year-old fossilized bacteria in South Africa. Then, in 1987, he and some colleagues reported that they had found the 3.465 billion-year-old microscopic fossils at a site called Warrawoona in the Western Australia outback, the ones he would display at the NASA press conference. The bacteria in the fossils were so sophisticated, Schopf says, that they indicate life was flourishing at that time, and thus life originated appreciably earlier than 3.5 billion years ago. Since then, scientists have developed other methods for detecting signs of early life on Earth. One involves measuring different isotopes or atomic forms of carbon. The ratio of the isotopes indicates that the carbon was once part of a living thing. In 1996, a team of researchers reported that they had found life's signature in rocks from Greenland, dating back 3.83 billion years. The signs of life in Australia and Greenland were remarkably old, especially considering that life probably could not have persisted on Earth for the planet's first few hundreds of millions of years. That's because asteroids were bombarding it, boiling the oceans, and likely sterilizing the planet's surface before about 3.8 billion years ago. The fossil evidence suggested that life emerged soon after our world cooled down. A quick start to life on Earth could mean that life could also emerge quickly on other worlds, either Earth-like planets circling other stars, or perhaps even other planets or moons in our own solar system. Of these, Mars has long looked the most promising. The surface of Mars today doesn't seem like the sort of place hospitable to life. It is dry and cold, plunging down as far as -220 degrees Fahrenheit. Its thin atmosphere cannot block ultraviolet radiation from space, which would devastate any known living thing on the surface of the planet. But Mars, which is as old as Earth, might have been more hospitable in the past. The gullies and dry lake beds that mark the planet indicate that water once flowed there. There's also reason to believe, astronomers say, that Mars' early atmosphere was rich enough in heat-trapping carbon dioxide to create a greenhouse effect, warming the surface. In other words, early Mars was a lot like early Earth. If Mars had been warm and wet for millions or even billions of years, life might have had enough time to emerge. When conditions on the surface of Mars turned nasty, life may have become extinct there. But fossils may have been left behind. It's even possible that life could have survived on Mars below the surface, judging from some microbes on Earth that thrive miles underground. When NASA's McKay presented his pictures of Martian fossils to the press that day in 1996, one of the millions of people who saw them on television was a young British environmental microbiologist named Andrew Steele. He had just earned a PhD at the University of Portsmouth, where he was studying bacterial biofilms that can absorb radioactivity from contaminated steel in nuclear facilities. An expert at microscopic images of microbes, Steele got McKay's telephone number from directory assistants and called him. "I can get you a better picture than that," he said, and convinced McKay to send him pieces of the meteorite. Steele's analyses were so good that soon he was working for NASA. Ironically, though, his work undercut NASA's evidence. Steele discovered that earthly bacteria had contaminated the Mars meteorite. Biofilms had formed and spread through cracks into its interior. Steele's results didn't disprove the Martian fossils outright. It's possible that the meteorite contains both Martian fossils and Antarctic contaminants. But, he says, the problem is, how do you tell the difference? At the same time, other scientists pointed out that non-living processes on Mars also could have created the globules and magnetite clumps that NASA scientists had held up as fossil evidence. But McKay stands by the hypothesis that his microfossils are from Mars, saying it is consistent as a package with a possible biological origin. Any alternative explanation must account for all of the evidence, he says, not just one piece at a time. In 2000, Oxford paleontologist Martin Brazier borrowed the original Warrawoona fossils from the Natural History Museum in London, and he and Steele and their colleagues have studied the chemistry and structure of the rocks. In 2002, they concluded that it was impossible to say whether the fossils were real, essentially subjecting Schopf's work to the same skepticism that Schopf had expressed about the fossils from Mars. The irony was not lost on me, says Steele. In particular, Schopf had proposed that his fossils were photosynthetic bacteria that captured sunlight in a shallow lagoon. But Brazier and Steele and co-workers concluded that the rocks had formed in hot water loaded with metals, perhaps around a superheated vent at the bottom of the ocean. Hardly the sort of place where a sun-loving microbe could thrive. And microscopic analysis of the rock, Steele says, was ambiguous, as he demonstrated one day in his lab by popping a slide from the Warrawoona chert under a microscope rigged to his computer. "What are we looking at there?" he asks, picking a squiggle at random on his screen. "Some ancient dirt that's been caught in a rock? Are we looking at life? Maybe. You can see how easily you can fool yourself. There's nothing to say that bacteria can't live in this, but there's nothing to say that you are looking at bacteria." Schopf has responded to Steele's criticism with new research of his own. Analyzing his samples further, he found that they were made of a form of carbon known as kerogen, which would be expected in the remains of bacteria. Of his critics, Schopf says, they would like to keep the debate alive, but the evidence is overwhelming. The disagreement is typical of the fast-moving field. Geologist Christopher Fido of George Washington University and geochronologist Martin Whitehouse of the Swedish Museum of Natural History have challenged the 3.83 billion-year-old molecular trace of light carbon from Greenland, saying the rock had formed from volcanic lava, which is much too hot for microbes to withstand. Other recent claims also are under assault. A year ago, a team of scientists made headlines with their report of tiny tunnels in 3.5 billion-year-old African rocks. The scientists argued that the tunnels were made by ancient bacteria around the time the rock formed. But Steele points out that bacteria might have dug those tunnels billions of years later. If you dated the London Underground that way, says Steele, you'd say it was 50 million years old, because that's how old the rocks are around it. Such debates may seem indecorous, but most scientists are happy to see them unfold. What this will do is get a lot of people to roll up their sleeves and look for more stuff, says MIT geologist John Grotzinger. To be sure, the debates are about subtleties in the fossil record, not about the existence of microbes long ago. Even a skeptic like Steele remains fairly confident that microbial biofilms lived 3.2 billion years ago. You can't miss them, Steele says of their distinctive web-like filaments visible under a microscope. And not even critics have challenged the latest from Minnick Rosing of the University of Copenhagen's Geological Museum, who has found the carbon isotope life signature in a sample of 3.7 billion-year-old rock from Greenland, the oldest undisputed evidence of life on Earth. At stake in these debates is not just the timing of life's early evolution, but the path it took. This past September, for example, Michael Tice and Donald Lowe of Stanford University reported on 3.416 billion-year-old mats of microbes preserved in rocks from South Africa. The microbes, they say, carried out photosynthesis but didn't produce oxygen in the process. A small number of bacterial species today do the same. Anoxygenic photosynthesis, it's called. And Tice and Lowe suggest that such microbes, rather than the conventionally photosynthetic ones studied by Schopf and others, flourished during the early evolution of life. Figuring out life's early chapters will tell scientists not only a great deal about the history of our planet, It will also guide their search for signs of life elsewhere in the universe, starting with Mars. In January 2004, the NASA rovers Spirit and Opportunity began rolling across the Martian landscape. Within a few weeks, Opportunity had found the best evidence yet that water once flowed on the planet's surface. The chemistry of rockets sampled from a plane called Meridiani Planum indicated that it had formed billions of years ago in a shallow, long-vanished sea. One of the most important results of the rover mission, says Grotzinger, a member of the rover science team, was the robot's observation that rocks on Meridiani Planum don't seem to have been crushed or cooked to the degree that Earth rocks of the same age have been. Their crystal structure and layering remain intact. A paleontologist couldn't ask for a better place to preserve a fossil for billions of years. The past year has brought a flurry of tantalizing reports. An orbiting probe and ground-based telescopes detected methane in the atmosphere of Mars. On Earth, microbes produce copious amounts of methane, although it can also be produced by volcanic activity or chemical reactions in the planet's crust. In February, reports raced through the media about a NASA study allegedly concluding that the Martian methane might have been produced by underground microbes. NASA headquarters quickly swooped in, perhaps worried about a repeat of the media frenzy surrounding the Martian meteorite, and declared that it had no direct data supporting claims for life on Mars. But just a few days later, European scientists announced that they had detected formaldehyde in the Martian atmosphere, another compound that, on Earth, is produced by living things. Shortly thereafter, researchers at the European Space Agency released images of the Elysium Plains, a region along Mars' equator. The texture of the landscape, they argued, shows that the area was a frozen ocean just a few million years ago, not long in geological time. A frozen sea may still be there today, buried under a layer of volcanic dust. While water has yet to be found on Mars's surface, some researchers studying Martian gullies say that the features may have been produced by underground aquifers, suggesting that water, and the life forms that require water, might be hidden below the surface. Andrew Steele is one of the scientists designing the next generation of equipment to probe for life on Mars. One tool he plans to export to Mars is called a microarray, a glass slide onto which different antibodies are attached. Each antibody recognizes and latches onto a specific molecule, and each dot of a particular antibody has been rigged to glow when it finds its molecular partner. Steele has preliminary evidence that the microarray can recognize phosphohopanes, molecules found in the cell walls of bacteria in the remains of a 25-million-year-old biofilm. This past September, Steele and his colleagues traveled to the rugged Arctic island of Svalbard, where they tested the tool in the area's extreme environment as a prelude to deploying it on Mars. As armed Norwegian guards kept a lookout for polar bears, the scientists spent hours sitting on chilly rocks, analyzing fragments of stone. The trip was a success. The microarray antibodies detected proteins made by hardy bacteria in the rock samples, and the scientists avoided becoming food for the bears. Steele is also working on a device called MASS, Modular Assays for Solar System Exploration, which is tentatively slated to fly on a 2011 European Space Agency expedition to Mars. He envisions the rover crushing rocks into powder, which can be placed into MASSI, which will analyze the molecules with a microarray, searching for biological molecules. Sooner, in 2009, NASA will launch the Mars Science Laboratory rover. It's designed to inspect the surface of rocks for peculiar textures left by biofilms. The Mars lab may also look for amino acids, the building blocks of proteins or other organic compounds. Finding such compounds wouldn't prove the existence of life on Mars, but it would bolster the case for it and spur NASA scientists to look more closely. Difficult as the Mars analyses will be, they're made even more complex by the threat of contamination. Mars has been visited by 9 spacecraft, from Mars 2, a Soviet probe that crashed into the planet in 1971, to NASA's Opportunity and Spirit. Any one of them might have carried hitchhiking Earth microbes. It might be that they crash-landed and liked it there, and then the wind could blow them all over the place, says Jan Toporski, a geologist at the University of Kiel in Germany. And the same interplanetary game of bumper cars that hurtled a piece of Mars to Earth might have showered pieces of Earth on Mars. If one of those terrestrial rocks was contaminated with microbes, the organisms might have survived on Mars, for a time at least, and left traces in the geology there. Still, scientists are confident they can develop tools to distinguish between imported Earth microbes and Martian ones. Finding signs of life on Mars is by no means the only goal. If you find a habitable environment and don't find it inhabited, then that tells you something, says Steele. If there is no life, then why is there no life? The answer leads to more questions. The first would be what makes life-abounding Earth so special. In the end, the effort being poured into detecting primitive life on Mars may prove its greatest worth right here at home. Thanks for watching. To stay updated on the latest discoveries, make sure to subscribe to my channel and turn on notifications.